My name is Lotta Ratu. I'm speaking on behalf of Milton E Biotech as the Global Product Manager for cell separation. And today we're going to be talking about how automation meets cell separation, primary cells for drug discovery assays. Today we're going to be covering Milten E Biotech in general very quickly as a pioneer in cell research and therapy. We'll take a look at density gradient centrifugation and cell separation directly from leukopak and Buffy code. We'll also take a look at automated cell isolation, and finally, the applications of primary cells in drug discovery assays. Milten E Biotech was founded in 1989 by Stefan Miltenyi. The basis of max cell isolation is the column technology that was developed as part of his diploma thesis and patented in 1990. Already in the beginning stages, Stefan Miltenyi had a pioneering spirit, and he saw the potential of max technology to become a revolutionizer in the field of gene therapy. The CliniMax CD34 reagent system is a method for selecting hematopoietic stem cells from donor apheresis while depleting T cells that can cause graft versus host disease. The development went on for over 20 years, including setting up a GMP manufacturing site in Teterow in Germany. And finally, CD34 isolation using the CliniMax together with the CD34 CliniMax reagent received FDA approval in 2014. Currently, the treatment of patients with AML is in first complete remission. So what started out as one man's diploma thesis has since grown into a global company of 2,500 employees worldwide and over 20,000 scientific publications citing max technology. Annually, more than 2,500 patients receive cell products manufactured with Milten E Biotech technologies. So what is max technology about? Well, there's 3 easy steps to the isolated cells of interest that you're going to want for your research and gene therapy. First step is the magnetic labeling, where an antibody conjugated to a tiny paramagnetic microbead binds to a receptor or protein on the cell surface. There are different strategies for doing this, as we'll see later. The second step is magnetic separation, where the cells are passed through a column that's positioned within a magnet. The cells labeled with the microbeads are retained in the column, while everything else is washed through in a gentle way. This flow-through is called the negative fraction, and it contains the cells without the ligand for the antibody or antibodies that were used for the labeling. In the elution step, the column is removed from the magnet, and this enables the magnetically labeled cells to be washed out. This can be done either manually, using a plunger, or automated, using a vacuum to pull the cells out, or in a fluidic system, just by washing at a higher speed. All of these 3 methods are gentle for the cells and separate the cells into the negative and positive fractions with exceptional purity and high viability. So the max technology is therefore based on 3 components. First, you have the microbead, you have the column, and the max magnetic separator. There are several versions of each of these available, depending on your starting material, cell type of interest, and whether you're using a manual or an automated system. And since the column is at the heart of max technology, let's take a quick look at why we use it and why it's so superior compared to other methods. The magnetic separators in column-based and column-free systems have a comparable magnetic force. The difference between the systems is the column. It amplifies the gradient of the magnetic force by 10,000-fold. It ensures that the magnetic field is constant throughout the column. So this minimal labeling of cells with tiny nanobeads is enough to retain the target cells in the column. The ion spheres have a cell-friendly coating. They enable free flow-through of the unlabeled cells through the column and thorough washing for removal of cell debris and contaminants. All of these 3 points enable the isolation of really pure and really high pure cells and high recoveries. Therefore, the max columns enable cell separation with minimal effects on cells for research and clinical applications. This translational approach from benchtop to bedside is reflected in all of our products. We provide the same products in research grade as well as GMP and clinical quality to ensure smooth transition into the clinic. We provide the entire workflow of products for cell-based work, from tissue dissociation and preservation to cell isolation and culture, including activation expansion, as well as cell analysis and imaging. When we talk about cells, we can choose between primary cells and cell lines. And before we get into the specifics of the primary cell isolation, let's consider the use of primary cells and cell lines in drug discovery. Primary cells are sourced by culturing cells derived directly from human or animal tissue. Cell lines, on the other hand, they're generated by immortalizing primary cells with vectors and culturing them over many passages. The cell cultures that result as this process goes on, they differ vastly with regards to resemblance to the tissue of origin, the characteristics, and the relevance to predict the actual biology from the tissue that they originate from. Primary cells are generally seen as limited in supply because they need to be fresh. Cell lines, on the other hand, can be virtually unlimited because you can culture them over long periods of time. Primary cells have a low proliferation rate in culture and therefore a limited time span. Primary cells have high donor variation, of course, because they result from primary tissues, whereas cell lines don't really have donor variation. This has been taken out. The mutation rate in primary cells is low as compared to cell lines where you have a high mutation rate, which means that you might have very different characteristics in the actual cell line that you have with regards to the primary tissue. And it might change also in the course of time while you're culturing the cells. The primary cells are therefore representative of the biological phenotype and complexity that you have in your original tissue. Cell lines, on the other hand, are generally seen as having a low relevance in vivo, and they're mainly used for focused research on a specific target. Primary cells are pre-characterized and ready to use. And for cell lines, of course, you need immortalization and authentication. And of course, there are these concerns of misidentified cell lines and cross-contaminated cell lines. I'm sure you're all familiar with these issues. So in summary, primary cells provide more relevant data for biological assays. How do we isolate these primary cells? Because, of course, somehow we need to come from the actual product to the culture that can be used in assays. So there are 4 steps that we need to take to go from primary cells to cells that you can actually use in assays. First, you have the starting material, sample preparation, max cell isolation, and the downstream application. And we'll go through all of these steps very quickly. One of the starting materials that you might have, or very commonly have, might be a blood product. And of course, here we're talking mainly about human samples. There are different kinds of blood products that you might be using: whole blood, buffy coat, leukopak, maybe cord blood, maybe bone marrow. You might also be using tissue that might be of human or mouse origin. And here we have healthy tissue, tumor tissue, and xenografts that can be planted into mice. What we normally still have to do is take the sample preparation steps. For blood products, this is mostly density gradient centrifugation to generate PBMCs, or for tissue, dissociation to generate a single cell suspension, for example, with the multimaxx dissociator. In the cell separation stage, of course, this we've already seen, you go through the magnetic cell isolation procedure to your population of interest. And here you have to consider different options for downstream applications. Are you going to be doing a positive or an untouched isolation? Do you need your epitopes free, for example, for a second sort or for flow cytometry? Would you like to work with or without phycol or a lysis of the cells? Would it make sense? Is there a direct approach that is possible, or will you need to go for an indirect separation? Are you going to be working with a lot of samples in parallel, or would you rather have sequential separations? And are you going to be isolating all of your cells at the same time, or are you going to be freezing, for example, PBMCs? All of these you should take into consideration when you're thinking about the different separation strategies. In terms of downstream applications, you might have, for example, an additional separation of different subpopulations, for example, Tregs, where you might want to go for CD4 followed by CD25. Another example is flow cytometry, where you might be wanting to look at immunophenotyping of cells, for example, TILs from tumor tissue. We also have cell culture applications, which are very widespread, of course, in drug discovery, where you could have functional in vitro assays, for example, killing assays. And, of course, a huge variety of different omics technologies. For example, RNA sequencing, single-cell sequencing, lots of different methods with which you might want to look at the cells on a molecular basis. We'll be looking at immunophenotyping and the functional assays in a little bit more detail in the applications part. But first, I'd like to draw your attention to the generation of PBMCs, which unfortunately for many is a must in the workflow towards cells for primary assays. There are two ways of isolating human blood cells. And PBMCs are, of course, the one that everybody knows best. Here you go from sample to PBMC to target cell. And here we have the two different options to isolate from your PBMCs: MAXX microbeads, where you will be labeling the target cell of interest in a direct way, or the MAXX isolation kits, where you will label everything that you're not interested in, leaving your target cells untouched. This workflow, however, requires you to go from sample to PBMC via density gradient centrifugation. I'm sure I don't need to tell you about density gradient centrifugation. Everybody knows how many steps it takes. Everybody knows how long this process takes. You have to layer the density gradient medium. You have to count your PBMCs, run a lot of washing steps in between, count your cells, and then you can proceed with the magnetic labeling. That's why we have introduced the hassle-free straight-from microbeads. They make the PBMC step completely unnecessary and solve all the problems that go together with phycol. There's no density gradient centrifugation. There's no cell counting. It's only a few simple steps, little hands-on and no user variation. And it's the fastest available protocol for target cells from Buffycoat and Leukopak. The protocol with which you would run the straight-from microbeads, this is specifically for Buffycoat. However, the protocol looks very similar for other types of straight-from protocols, for example, Leukopaks. So you take up to 80 milliliters of your Buffycoat, or if it's less, fill up to 80 milliliters. Add the beads, incubate for 15 minutes in the fridge, and then proceed to separate the cells over 12 columns on, for example, the multimaxx. The whole process for a Buffycoat takes about 25 to 30 minutes without density gradient centrifugation, no lysis, and no cell counting. And this is for Buffycoat. For Leukopak, you would be looking at about 60 minutes. Therefore, we can now fill in the other side of this table, where we go directly from sample to your target cell without density gradient centrifugation or the PBMC step in between. We've brought out the straight-from microbeads. Whole blood was available already a few years ago, and over the last year, we've brought out the Buffycoat, LRSC, and Leukopak microbeads. And again, the straight-from microbeads enable you to go directly from sample to target cell without any steps in between. So to summarize so far, we've now learned about primary cells, what we need to consider when planning a project isolating cells, the steps to isolate them from tissue or blood products, as well as the shortcuts that we can take, for example, straight from. Everything we've talked about aims at generating assay results that are biologically relevant, reproducible, and as efficient as possible, saving hands-on time and unnecessary steps and repeat work in the lab. But the reagents we work with are only one half of the solution. Truly efficient and reproducible protocols require automation. So I'd like to take a few moments to discuss automated solutions before going on to discuss the drug discovery assays. So why is it that we need automation? As we've already said, automation reduces hands-on time and frees you to work on more important tasks that cannot be automated. It minimizes variation between different users in the lab, or even good and bad days of an individual scientist. And of course, it minimizes the costly human errors. With standard protocols and minimum user variation, you can be sure that the effects that you're seeing are due to your assay results rather than artifacts. In short, the automation reduces redundant experiments and allows you to focus your time on where it matters. And very shortly, I'd like to introduce the Milten E Biotech automated solutions for magnetic cell isolation, and then we'll get to the drug discovery applications. First, we have the multimaxx Pro separator. It's an easy-to-use, intuitive, first automated instrument for a multi-user lab. You have sequential separation of 1 to 6 samples per run, and a large variety of different sample types and applications that you can mix and match even within one run. You have true walk-away automation with automated magnetic labeling and separation. You also have reusable columns, so you can use the columns for 14 days in this instrument compared to other methods where we only have single-use columns. And you have preset programs for high purity and high recovery, depending on what you need from your cells. So when we talk about the multimaxx, it's sequential and fairly low throughput. When we talk about the multimaxx on the other side, we have parallel cell separation of large sample numbers and high sample volumes. The instrument is semi-automated, but it's scalable. So you can go from 1 sample to 24 samples in one run, and it's compatible with all of our manual columns, LS, LD, and whole blood columns. And we also have a multi-24 column block, which enables you to process 24 samples in one sitting without having to work with individual columns. You can use the 24 magnets as individual magnets for small samples, as well as just one for large volume samples, such as buffy coats and leukoplaques, which makes the instrument really flexible. However, it's still only semi-automated, and you have several manual pipetting steps. If you'd like to go for a fully automated high throughput option, you would want to look at the multimaxx X, which we've only just launched. You have fully automated sample and buffer handling and magnetic labeling, and you can separate cells from an entire buffy coat in less than 30 minutes or a leukoplaque in 60 minutes. You can also go, for example, from 24 mouse tumor samples that can be processed in about 50 minutes to an hour. And all of the programs are tailored to ensure the perfect fit into your protocols, including the preparation of downstream applications. Taking a quick look at the multimaxx X, the heart of the multimaxx X is the multimaxx Cell 24 instrument, which we already saw before, and everything else is built around the multimaxx Cell 24 to service that, the processing of the cells. We have the elution station, which enables the vacuum impulse that elutes the positively labeled cells, and the elution chamber in which this happens. Samples and reagents are kept cold throughout the entire time, and you can have your samples in 15 milliliter or 50 milliliter racks, depending on the volume. In the back, we have storage hotels that enable storing of deep well plates or pipette pips. And of course, we have the buffer troughs, disposable tips, as well as the columns that we use for the actual isolation. So with this, I'd like to get to the summary of in the instrumentation part. As we've seen, the multimaxx Pro is a very intuitive, easy-to-use instrument for low to medium throughput. You can run 1 to 6 samples per run and go for up to a medium kind of sample volume if you're talking about blood products. However, it is a fully automated instrument and a really good fit for a lot of labs. Because of these features, it's perfect for small PBMC aliquots, or for mouse samples and patient-derived xenografts. Because it can process whole blood directly as whole blood straight from, it's also really good for patient and donor samples where you don't have large volumes. When we compare it with the multimaxx, again, we have full automation. The sample volume, if we're talking about blood products, is the maximum kind of throughput that we can have there. And you can go from 1 to 24 samples in one run. The multimaxx Cell 24, very similar, however, of course, only very low automation here. These features enable the multimaxx family to isolate small human samples in parallel, up to 24, really good for that, up to 24 mouse samples in parallel, and as we've already discussed, large human samples. So here you can kind of see where these instruments fit and what the benefits are. So we've now learned about the steps of primary cell isolation and how to ensure maximum efficiency and reproducibility by selecting the best reagents and instruments to match your workflow. And at this point, I'd like to take the chance to go through some of the application data from assays that are frequently run in drug discovery at different stages. We have phenotypic screening, we have for target discovery, we have animal model development, toxicity or immunogenicity, and we'll start with phenotypic or high-content screening. Phenotypic screening can be used for biologics and small molecule development. The example here is small molecules, where we looked at the binding of CCR2 on the surface of monocytes, so isolation of monocytes here. For high-content screening in the development of an anti-inflammatory drug, a CCR2 inhibitor. CCR2 is expressed on monocytes, as you can see here, and it's the binding site for MCP1, a chemokine that attracts and activates monocytes. In the top row here, you can see the blue monocytes. I hope you can see also the red in the back. In the top row, you can see the blue monocytes on the left, the red MCP1 molecules in the middle, and in the overlay here, you can see that they are co-localized in the same place. In the middle row, unlabeled MCP1 was added in excess to block all the binding sites on the monocytes. And as you can see, there is no labeled MCP1 that is able to bind because all of the binding sites are blocked. And exactly the same can be seen when you add CCR2 here, and you have no free binding sites, which means that you have been able to block the reaction using this drug candidate. You can then get additional information at a single cell level, for example, on cytoplasm shape, size and area, and some information on the nucleus. In the later stages of the drug discovery process, you'll be needing lots of primary cells for the mechanism of action studies for your drug candidates to understand, for example, the exact mechanisms by which the drug influences the immune system, which concentrations are needed, and in what kind of combinations you'd like to be using it. In the example of an MLR assay, you'll isolate the monocytes from one donor and the T cells from another one, and co-culture the matured dendritic cells with the T cells, leading to activation of the T cells. Adding then a checkpoint inhibitor molecule unleashes the brakes on the immune system and enhances the reaction. The T cell activation can be tracked, for example, by interferon gamma production, as you can see here. So the controls and the T cells by themselves show low basis activation or interferon gamma production. However, when you add a PD-1 antibody, in orange or PD-L1 antibody in purple, you can see significantly increased interferon gamma production in all of the three donor pairs as a sign of drug candidate and enhancing the immune reaction. Another method of MOA profiling is the Treg suppression assay. Here you isolate the T cells and stimulate them, and then add the Tregs and co-culture them. During the co-culture, you can track the inhibition of the T cell activation by the Tregs. And by adding the drug, you can detect the different compounds that can inhibit the Treg inhibitory effect, as you can see here. So you can compare the different drug effects in the MOR assay. The same method can also be used, for example, in the MDSC suppression assay, which is based on the same principles. Finally, we have the TDCC assay. Here, you isolate the pan-T cells and culture them with antigen positive, in this case EGFR positive, cancer cells. In this case, we have a CD3 EGFR bispecific antibody that brings the T cells and cancer cells together for more efficient killing. This can be then monitored on an ATP-based readout, as you can see here. So this example shows that the EGFR CD3 antibody specifically kills the cancer cells at different concentrations, compared to the control antibody that doesn't have the EGFR specificity. Similarly, you can use cell lines expressing firefly luciferase reporter gene, as you can see here on the right. So of course, you can use different kinds of methods to run a very similar assay as well. So this was our last example on MOA, but of course we have many more. So if you don't see data for your specific assay, just ask us, and we're very much prepared to help you with any of the assays that you're running. In the later stages of drug discovery, you'll be looking at predictive immunogenicity. You might be wanting to use the MAPS assay, for example. In this assay, you isolate the monocytes and differentiate them to immature DCs, and then you add the biologic drug candidate. You can precipitate the bound fragments that are presented on the DCs and analyze which peptides are presented by mass spectrometry, and then identify the ones which might cause an immunogenic reaction on the T cells. The result of this is a mass spectrometry output that might look something like this. Here we have an example of an antibody, the heavy chain and the light chain of a therapeutic antibody. And you list all of your donors and map the different peptides that were presented in a heat map for your specific antibody. The heat map can then be used to modify the antibody to de-immunize it and make it safer with regards to immunogenicity. When you have this data, you might want to use it for a DCT assay, which would be the next step in predictive immunogenicity. Here you isolate the monocytes that then act as antigen-presenting cells and co-culture them with the T cells. You add the candidate antibody either as a whole or the peptide mix that you found from the MAPS assay. And then you see if the CD4 positive T cells proliferate and produce IL-2, that indicates an immunogenic reaction to the peptides. This can then be tracked using flow cytometry by measuring the CD4 proliferation and IL-2 production using a cytokine release assay. And here on the right, you can see that the correlation between clinical data and the in vitro data shows that if a drug shows immunogenic effects in the in vitro studies, it's highly likely to cause problems in the patient as well. In the last example, we're looking at immunophenotyping. So when you take a bulk tumor and run it through a flow cytometer, the tumor cells are going to be outnumbered the immune cells are going to be outnumbered by the tumor cells, so that you're not going to get statistically relevant data in your plots. As you can see here, even CD8, I'm not sure if you can see it in the back, but CD8 and CD19 are barely visible. And, for example, rarer populations, such as tumor-associated macrophages, are barely seen at all down here. However, when you dissociate your tumor, use the cell separation TIL microbeads to pre-enrich for the tumor-infiltrating lymphocytes, the whole picture looks completely different. You have that statistically relevant data in your plots, which ensures that you can actually trust your results and you can say, this is actually the effect that I'm seeing. This can be used, of course, for many different things. We have just released the TIL microbeads, CD45, 4s and 8s, for human and mouse tumors. And so this would be a perfect example of where these kind of rare populations can be isolated in a really good way. This brings me to my summary. We've now seen that primary cells are a valuable model for biologically relevant assay results. We've also seen that reliable results depend on the right isolation strategies, reagents, and instrumentation, and that automation can reduce human error and improve efficiency with automated solutions in every stage of drug discovery.